The membrane of cells that are involved in absorptions have hair-like extensions or projections we call microvilli. Look at them, microvilli. See these little things going up and down like that? Microvilli. Microvilli. They are found in the small intestine where we absorb food particles into the bloodstream. Microvilli extend the cell surface, creating a bigger surface or area for absorptions. They also allow for enzymes to reside there and attach, which then involve in breaking down polymers into absorbable monomers, disaccharides into monosaccharides, for example. We call these brush border enzymes as well. These enzymes, they're brush border enzymes. We call them that because under a microscope they look fuzzy like a little brush. The immune system of people with celiac disease is triggered by the gluten in the protein in wheat, rye and barley. Immune cells then destroy their own microvilli in an autoimmune response. You can only imagine how that gives us trouble because absorption leading to malnutrition. It also gives cramping and diarrhea. Not pleasant. Celiac disease cases are on the rise. It is often thought that the major causes of breeding increased the percentage of gluten in the wheat. However, my research has not found that to be true. I personally believe we just eat a lot of it, which over time makes us more reactive and vulnerable to it. I simply try to listen to my body if I can, and it keeps telling me to eat a little bit less spready stuff and not as much. Everything in moderation, I guess. The inside of the cell is a gel-like substance made of 70 to 80% of water with proteins suspended in it. It's called the cytosol. Cytosol. And it is made of glucose, amino and fatty acids and other particles that are floating in it. There are also little structures with specific functions that we get to shortly, and those, we call those organelles. Organelles. Look at all these things over here. These are all the little organelles. And then centrally located is a dark stained thing here. See this dark stain? That's the nucleus, and it houses the DNA and the double helix. So that's the nucleus in there. And now, adding the cytosol plus the organelles, but not the nucleus, gives us the cytoplasm. And we see that over here, right here, cytoplasm. A network of protein fibers help maintain the cell's shape and do some other pretty cool things. Collectively, we call them the cytoskeleton. Cytoskeleton. They are made of three main types of fiber. Microfilaments are the narrowest. They are two intertwined strands of protein beads called actin. See here the actin subunits. These actin filaments assemble and disassemble quickly and serve as tracks, a movement for proteins, motor proteins that then we call myosin. Actin filaments are very, very useful in cellular motion. So they're good for cellular motion. White blood cells, for example, use them to move themselves on the sides of potential infections. So they go and glide through the blood vessels or the blood walls, the cell walls, and then go through the tissues. And they do that with the assembling and disassembling of these structures. And we call those amoeboid motions when they do those. Those are amoeboid motions. Boid motions or movements. It's pretty cool stuff. Or actin and myosin filament interactions are also on a super large scale what actually causes muscle contractions. as well. Microfilaments also give us this rigidity to the cell's form, but a much bigger player in that is the intermediate filaments, or the intermediate filaments. Intermediate filaments. And those are proteins wound together, protein strands wound together, as we can see, and they create a lot of structural support to the cell. They also help the nucleus and other organelles in place. They also do that. Their protein types are very diverse. Some of them are, for example, keratin. Keratin is one of those proteins. And we know keratin from our skin layer. It makes it tough, it makes it leathery and waterproof. And those are intermediate filaments. So they're plentiful also in hairs and in nails, wherever we got toughness going on. Microtubules are the largest one of them, although they're small, of course, and they are hollow kind of structures. They're like tubules like that. And what they do is they help the cell's resistance to compressive forces. And so they make sure that the cell doesn't collapse. They make me think like railroad tracks, because what they also do is they have molecules walk on them and move throughout the cells. They are kind of like the first ones, the microfilaments. They can assemble and disassemble very fast and quickly. Microtubules, for example, are used during the cell division when the copied genetic DNA is pulled apart to each of the cell's poles, so to speak, sides, and then you have the genetic material evenly distributed in each offspring and not unevenly distributed, which is very important. They're also what creates long hair-like extensions from the plasma membrane that act like whips. We're going to talk about those next. Here are some of those hairline extensions. When there are many, they move or whip substances past the cell. You can see them here, how they do that. They whip like this over here. They whip substances against the cell. Those are known as cilia. And if there is only one or a couple of them, those are flagellum. And what the flagellum do is they move a whole cell forward when they move in propeller-like motion like that. Both extensions are made of microtubules and looking like that kind of structure over here. Look at this picture. It shows how microfilaments, right in here on the outside, resist tension in the cortex, microfilaments. And like rubber bands, they compress a little and they resist the tension coming from the inside. And then microtubules, they're right here, microtubules, internally resist that compressive force and push outwardly. And then intermediate filaments hold the organelles in place. Very interesting, very cool stuff. It's fascinating to me how organisms use relatively simple, ingenious structures such as the phospholipids forever applicable. The phospholipid bilayer are arranged in sheaths with the hydrophobic tails pointing to the inside. Invert makes that oil film right in here. But we can also use, instead of a bilayer, use a simple layer when we can then use to travel in a sphere, travel lipids around in the body. It's just very ingenious. Inside the cell, the phospholipid bilayer surrounds the genetic material and becomes the nucleus. The nuclear envelope, right here, is actually two layers of a double lipid bilayer to really protect that inside stuff, because the DNA is extremely sacred. And then we got nuclear pores right in here, nuclear pores, that within the cell membrane, and then they control what goes in and out of the nucleus. Much of these internal phospholipid sheets connect to the nucleus and create a series of connecting sacs and tubules. And we call those collectively the endomembranous system. Endomembranous system. Endo means inside, within. right in there. It is, this is where proteins and other molecules are made and then transported to where they're needed. When talking about the cell's organelles and their different tasks, I like to think of the whole cells as a village that needs to survive independently. What does it all need to do? It needs a power plant to make energy for sure, so everything can run. Well, we'll talk about that in a little bit. We also need factories that make stuff. And then we need a post office that packages the things and ships it out of the cell. Look at the endomembranous system. It's a continuous assembly line from the nucleus which holds the instruction to make proteins to then a manufacturing part of the system. And then more peripherally gets to the packaging area and where we ship it out into the cell or the outside world depending on where it needs to go. The endoplasmic reticulum comes in two forms. It comes in rough ER, as we call it, or in smooth ER as well. So we got here rough ER right here, rough ER, and over here we got some smooth ER as well. Oops, there we go. The rough looks dotted under the microscopes. Those dots are actually ribosomes which assemble the protein. We'll be talking about those next. The other one, the smooth one over here, doesn't have those dots on it, and they synthesize other molecules such as lipids and carbohydrates. Then further out, the Golgi apparatus right in here, also known as the Golgi body, right in there. A bunch of flat bags of membrane, you know, those phospholipid bilayer things. Here we go. It literally packages, it's a post office, it packages, it sorts, it tags, and ships the product from the ER. In phospholipids, you guessed it, the membrane is so fluid it can simply pinch off and form again and do all of that the way it wants. The Golgi then distributes those particles, those created things, to the inside of the cell where it needs to go, but also to the outside of the cell. See here, you can see how the cell membrane fuses and puts it out. That's kind of cool. In plants, the Golgi also synthesizes or makes polysaccharides, and some of which then acts as chitins that are incorporated into the cell wall.